seed germination, right? See, in this chapter, this is one of the important topic from the seed germination or the seed germination inhibition process. There is nothing but some of the phytohormones are act as inhibitors, right? From this topic, definitely one bit is given. Right? This is a very important concept. So we know the seed germination process. When the seed have favorable conditions, that time the seeds produces some small external growths, which are nothing but very minute protruded structures. Okay, that tissue is nothing but one of the embryonic tissue. The embryonic tissue extended and forms there is a shoot pod and the root pod. We know which forms the plumule. The plumule is developed in the shoot pod, and there is a root pod in the root pod is developing from the radicle. It's developed from the radicle. So here the seed generation process, especially controlled by the two factors are there. One is called as external factors and other one is called as internal factors. Moreover, both the factors are very important for the seed germination process. So here external factors are there. We know the seeds, the very important external factor. The seeds germination process, the very important external factor is the water. So what is the very important agent for the seed germination process? Because we know when seed germination takes place, that time whatever the storage food is there, the storage food mobilization process, activation of enzymes, right, which are majorly required. So whatever the gibberellic acids are there, the gibberellic acids are responsible for the mobilization of storage food material, right. See, here we are discussing about the water, right. So here the water is the very important agent. First of all, in the dry seed, 6 to 15% of water is present. 6 to 15% of the water is present. And when the seeds absorbs the water, that is nothing but before the seed generation process, the seeds absorbs the water and all enzymes are active, presence of water, and here the percentage of water is 75 to 95 percentage. 75 to 95 percentage of the water, right, present within the seed. So dry seed. So why dry? Why seeds are dried? See, when the seeds are dried, which are easily transferred, that is nothing but see, whatever the seeds are there, the seed dispersal is very easy when these are having light weight. When the seeds are having light weight, right? So here the seed, germinated seed, 75 to 95% of the water, and softening of seed coat, presence of water, the seed coat is softened, then the seeds are easily germinated, and permeability of the seed is nothing but absorption of water. So whatever the seeds are there, the seeds absorbs the water, right? See, permeability of the seed, that one is supporting for the softening of the cell wall, that is nothing but softening of the seed coat, and transportation of gases. See, when the seeds are germinating, that time, okay, we discussed there is a plumule pod formation, radical pod formation from the embryonic tissue. So how the embryonic tissue produces these pods by the cell division process, by the mitosis process, these pods are produced. We know when the cell divisions takes place, that time energy is very important agent. The energy is produced presence of oxygen. So the concept is here, gases exchanges also takes place. So transportation of gases. So here the water is there. The water supporting for the softening of seed coat, and the permeability of the seed increases the absorption of nutrients and minerals from the soil, and the transportation of gases. We know very minute pore-like structure is there. Seed pore. The seed pore is completely opened, right? When the seed structure is bulged, the seed pore size is also increased, right? Hydrolysis of storage food material. When the seed contain food is there, presence of water, the food is nothing but storage food, presence of water, hydrolysis takes place, then the cell division takes place. Okay, presence of oxygen, energy is produced, more cell division takes place. So the water is supporting for the cell division process. And next is the temperature. See, some seeds are grown at the low temperature, some seeds are grown at the medium, right? Some are grown at the high temperature. So here the causal range is here. Okay, there is a 5 to 40 degrees centigrade. This is the common range, right? There is an optimum condition is also there. Optimum temperature is also there. Here the 25 to 30 degrees centigrade. 25 to 30 degrees centigrade. See, majority of the enzymes are grown at the 25 to 30 degrees centigrade because enzymes are very active. Enzymes are very active. Right? So that is about the temperature role within the seed germination process. And next is the oxygen. Presence of oxygen. Majority of the seeds are grown. Majority of the seeds are produces the seedling. That is nothing but the germination process. So here, majority of the seeds are germinated with the presence of oxygen because just now I said oxygen is very important agent for the production of energy, presence of energy, the cell divisions are takes place. So that one is nothing but the seedling process or the seed germination process. Right? Say there is a rice and typha, these are the two plants, okay, which are not grown presence of oxygen, which are not grown presence of oxygen. See, we know when before showing the rice seeds, okay, farmers did the ploughing. Farmers did the ploughing. When the ploughing is occurred with the soil, that time, see, a minimum amount of oxygen present within the that soil, right? See, when the rice seeds are shown, so whatever that ploughed, okay, whatever that ploughed soil is there within the inner side, within the inner sides of the soil, means wherever that ploughed soil is there, okay, somewhat small openings are there. So within that opening parts, within the ploughed region, okay, the seeds are covered. The seeds are covered by the soil. So excess amount of oxygen is distributed. Very minimum limited, oxygen is present. So here also oxygen is required, but not much oxygen. Very limited oxygen is required. So presence of the normal oxygen condition, these seeds are not grown. Mostly the enzymes are negative presence of oxygen. Much oxygen. Right? See, coming to the light, see there is a non-photoblastic seeds are there, photoblastic seeds are there. We know the concept is photoblastic. See, photo indicates here presence of light. Non-photoblastic. See, some of the seeds, the seed growth or the seed germination not influenced by the light. See, these seeds grow absence of the light, presence of the light also. These seeds can grow absence of the light and presence of the light also. So light is not affected to seed germination in some of the plants. Example is the maize and the beans. So within the maize and the beans, the plants, okay, the seed germination process not affected by the light. Right? And photoblastic means the seed germination affected by the light. Negative photoblastic means which are grown absence of the light. So absence of the light only these seeds are grown. Onion is the best example, lily is also one of the best example, right? And some of the seeds are grown presence of the light. See, there is in lettuce, we know that lettuce plant. See, the seeds having some of that photochrome pigments are there. The seeds contains that some of the photochrome pigments are there. The pigments are showing sensitive towards the light. So presence of the pigment, presence of the light. Some are born absence of the light, and some are born presence of the light. Right? So these are negative photoblastic and these are positive photoblastic. Right? That is about the effect of the light. And next one is that other factors. See, pH also influences the seed germination process. Some of the seeds are grown in acidic media. Some of the seeds are grown in alkaline pH. Most of the seeds are grown in the neutral pH. What we are seeing that majority of the plants grown within the neutral pH, but some are grown in acidic medium. Maybe the acidic soils are required. Some are grown in alkaline medium. See, phytohormones is the best example. These seeds are grown in presence of acidic soils, and see alkaline medium. Most of the garden plants or ornamental plants, most of the garden plants and some of the ornamental plants grown within the alkaline medium, right? And the last one is the obligatory parasites. See, we know some of the plants are the parasites. Okay, the parasitic plants also produce seeds. Okay, again when the seeds are germinated, when the host is available, that time is only these seeds are germinated. When the host is available, that time only these seeds are germinated. So we know that parasitic plant is the Varabanki, right? See, obligatory, compulsory, right? Compulsory host and parasite interaction. Host and here parasite interaction. So Varabanki is one of that the parasitic plant. These plants grow or the seeds are germinated presence of host plants only. If host plants are not available, the seeds are not germinated. Right? These are the some of the external factors. Next one we will discuss about the internal factors. Right? Coming to the next one is the internal factors. Right? See, these are present inside the seed and responsible for the seed germination process. All of you remember, most of the hormones are majorly controls the hormones, means here the phytohormones controls the seeding process or the seed germination process. See here, the very first is the maturity of the embryo. See, whatever the seeds are there, the seeds are produced, right? But majority of the seeds are not immediately germinated. See, for the seed germination process, that kind of seeds are required some time, right? After that time period, the seeds are germinated. So what happens? Whenever the seeds are produced, maybe at that time the seeds contains immature embryos, means which are not completely developed. So the immature embryos taken certain time period, after that or within that time period the cell divisions are takes place, then the immature embryos modified into the mature embryos. The immature embryos are modified into mature embryos by the cell division process, right? After that cell division process, that is nothing but mature embryos. Once the mature embryos are formed, then that embryos are ready to germinate. So the embryo condition is also very important here. The embryo condition is also very important. See, whatever the immature embryos at the time of the seed shading, that is nothing but the fruits are fall out, the seeds are released, right? The seeds releasing process, that is nothing but shade, right? So certain time period is taken, then which are modified into mature state, then which are ready to germinate. And come to the after the ripening. See, same here I mentioned here the immature embryos. Here I mentioned hormones are not available. After that seed shading process, the seeds not contains the phytohormones, or the seeds not produces the phytohormones. Immediately some of the seeds not produces the phytohormones. So phytohormones are not available, no cell divisions and no elongation process and no seed germination. So when these are available, when the phytohormones are available, the seed germination will be takes place. And viability. Remember, viability is nothing but the lifespan of the embryo. See, especially the seed coat is impermeable to water and oxygen, right? Initially impermeable, but once the seed coat is softened, in that case, water molecules are entered. Once the water is available, seed coats are softened. Then air, that is nothing but gas, is also entered. So here, observe the embryo. The embryo survives few weeks. So here, mostly the seeds are germinated within a few weeks. And the trifolium, that embryo viability is up to 100 years. So the seeds are taken up to 100 years time. Up to 100 years time. Right? That is about the trifolium. And lotus up to 1000 years. See, if the embryo have more viability for the seed germination, that seeds taken more time to germinate. Embryo have more viability, the seeds are taken more time to germinate. So oxal is few weeks, trifolium 100 years, and the lotus 1000 years. So that is about the viability of the embryo condition. Viability of the embryo condition. Dormancy factors, we know that hard seed coat, seed having the hard seed coat, and the growth inhibitors are there. Abscisic acid is the best example. See, we discussed inside the seed, abscisic acid is present. Outside unfavorable conditions are there. Then the abscisic acid inhibits the seed germination. So some of the growth inhibitor phytohormones are there. Ethylene is also right, and here abscisic acid also inhibitor. So high temperature and high concentration of carbon dioxide within the atmosphere, high concentration of carbon dioxide within the atmosphere, or if in certain cases inside the seed, okay, there is no permeability of the gases, okay, in that case carbon dioxide is accumulated. So in that case also the seeds are not willing to germinate. So that is about the dormancy condition. Remember here the seed coat is the major one, and next one is the growth inhibitors are the second one. Okay? And other factors. Okay, within the seed, within the embryo storage coat not available, the nutrients are not available, then the seeds are not willing to germinate, are not ready to germinate. When the seeds absorb the nutrients from the soil, when the embryo having all the types of the nutrients, that time the seeds are, that kind of seeds are germinated. Right? These are the internal factors regarding to the seed germination process. And next one is we discuss about the seed dormancy. This is about the seed germination, and we discuss about the dormancy also. Coming to the next one is the seed dormancy, avoiding process. What is seed dormancy? The seed having all favorable conditions outside the seed, all favorable conditions are there. Even though the seeds are not germinating, even though the seeds are not willing to germinate. So that condition is called as seed dormancy. Remember, whatever that the seed conditions are there, the seed conditions are okay. Means I am not talking about here external conditions. I am talking about the internal conditions. But even internal conditions also favorable, external conditions also favorable. And remember, whatever the seed coat is there, right? The seed coat is moreover inhibiting here seed germination. Moreover, the seed coat inhibiting the seed germination. Remember, the seed dormancy is nothing but the factors which are required for the seed germination. All these are available, but the seeds are not willing to germinate. Not willing to germinate. Simple. Whatever the conditions are required for the seed germination, all these are maintained, even though the seeds are not germinating because of some of the factors, influences. Remember, the very first one. Okay, see we are doing that avoiding the seed dormancy by certain methods. Here we are doing certain methods to avoid the seed dormancy. The very first step is called as scarification. We know scars. This is the seed having this thick seed coat, and we are generating some of the scars. Upon the seed coat, we are generating some of the scars or some of the perforations. We are generating some of the perforations by the mechanical method. Simply we are collecting the seed and we are applying that mechanical pressure. So whatever the hard seed coat is present here, the hard seed coat, that is the thick seed coat. The thick seed we apply the mechanical pressure, the hard seed coat or the thick seed coat change into the thin seed coat at certain regions. Where we apply the mechanical pressure, at that region the thick seed coat becomes thin seed coat. So this is one of the seed and the thick seed coat by the application of mechanical pressure or mechanical method, right? Whatever that hard seed coat is there at some regions, it becomes thin seed coat. At some regions it becomes thin seed coat because we are applying here mechanical pressure. Then water molecules are easily entered. Right? So once the water molecules are entered, the seeds are germinated. So simple one is the scarification. We are making the scars on the seed coat. By the mechanical pressure. And next one is the stratification. Stratification indicates here. This is also called pre-chilled treatment. The seeds are exposed to low temperature. This is one of the non-mechanical method. Seeds are exposed to low temperature. Then the seeds are germinated. Examples are here: apple, cherry, walnut, and polygon. Some of the seeds are there. This is the non-mechanical method. The seeds exposed to low temperature, right? That is nothing but 5 degrees centigrade. The seeds are exposed to the 5 degrees centigrade. Sometimes here temperature is also given 5 degrees centigrade or below the 5 degrees centigrade. In that case, the seeds are germinated. Right? That is the stratification. Last one is the chemical treatment. The seeds are treated with different types of chemical agents. One is the potassium nitrate. See, which are act on the seed coat and which are activates that storage food material. Gibberellic acid, which are promoting the seed germination process. Thiourea act on the thick seed coats. Then the seed coat is destructive. So some of the chemical agents are acting on the cell. Whatever that seed coat is there, some of the chemical agents are acting on the seed coat. And some of the chemical compounds like gibberellins which are act on the seed coat and the endosperm. That is what they call food fertilization. Then the seeds are germinated. So that is about the chemical treatment. So we discussed about factors affecting seed germination, and we are overcoming the seeds are overcoming the seed dormancy by the different types of methods. One is the mechanical method, other one is the non-mechanical method, the third one is the chemical method. Right? In this chapter, only one topic is there, right? Induction of flowering and different types of flowering plants. We'll discuss now. Maybe we'll discuss the next class. Then this chapter will be completed. Right? Phytohormone chapter will be completed.